Chromosome mapping. Definition, types, importance. Part 1. UGC-NET Anthropology. Welcome to AnthroNet Academy, a study aligned with the UGC-NET Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in Anthropology, complete with author names and insights important for UGC-NET exam. Definition. Chromosome mapping means determining the relative positions of genes in a chromosome by creating maps that are used to organize and understand genetic information on chromosomes. These maps show the positions of genes and the distances between them based on a specific scale. Chromosome mapping helps to understand the genome's organization, structure, and function. Thomas Hunt Morgan made significant contributions to the modern understanding of chromosome mapping. His experiments with fruit flies provided evidence for genetic linkage. Morgan's discovery that the white-eye gene in Drosophila was located on the X chromosome led to the identification and mapping of other X-linked genes. The resulting chromosome map revealed a linear arrangement of genes. Types of chromosome mapping. There are two main types of chromosome mapping techniques. 1. Genetic mapping. 2. Physical mapping. 1. Genetic mapping. Genetic mapping, also known as linkage mapping, refers to the method used to create genetic maps to estimate the positions of genes in chromosomes. These genetic maps are constructed by analyzing recombination patterns that occur during the crossing over of chromosomes. Genetic mapping starts by using linkage analysis to study the frequency of recombination between genes. Recombination frequency indicates genetic linkage and helps to determine whether the genes are linked together or not. A centimorgan, cm, is a unit that describes a recombination frequency of 1%. As the distance between two genes increases, the likelihood of recombination occurring also increases, resulting in a higher recombination frequency between them. The recombination frequency is determined by using the number of recombinant individuals compared to the total number of offspring. This frequency is expressed as a percentage. It is important to understand the concept of genetic linkage and recombination for genetic mapping. Genetic linkage and recombination. Mendel's law of independent assortment states that pairs of alleles segregate independently. However, upon the rediscovery of Mendel's work, it was known that some pairs of genes were inherited together due to their location on the same chromosome. This phenomenon is known as genetic linkage. Linked genes are located close to each other on a chromosome, which increases the chance of them being inherited together. So, the concept of linkage is used to understand the relative positions of genes on a chromosome. However, not all genes on a chromosome are necessarily linked. Genes that are located further apart from each other have a higher chance of being separated during recombination. Recombination occurs during meiosis, where homologous chromosomes pair up to form bivalents, and crossing over can occur between chromatids, leading to the exchange of genetic material. Genes that are closer together have a lower chance of crossover. Thus, the crossover percentage reflects the distance between the genes. Molecular markers for genetic mapping. A molecular marker is a specific DNA sequence that shows variation and can be identified using molecular techniques. It is used in mapping to locate its position on the genome. To map molecular markers, their inheritance patterns are observed in subsequent generations using techniques like Southern hybridization and PCR. Data is collected from the analysis of molecular markers, including information about the presence or absence of specific markers. Using specialized software, the collected data is processed to construct a genetic map that shows the relative positions of markers on the genome. Important molecular markers. 1. Restriction fragment length polymorphisms, RFLPs, are one of the earliest molecular markers used for genetic mapping. RFLPs are specific patterns of DNA fragments created by restriction enzymes that identify and cut DNA at specific sites. In genomic DNA, there can be variations in these restriction sites, leading to differences in the restriction patterns between closely related genomes. RFLP can be mapped on a genome by studying its inheritance pattern. 2. Simple sequence length polymorphisms, SSLPs, are DNA markers that consist of repetitive sequences with variations in the number of repeats. There are two types of SSLPs, mini-satellites and microsatellites. Mini-satellites are also known as the variable number of tandem repeats, VNTRs, and they have longer repeat units, up to 25 base pairs. Microsatellites, also known as simple tandem repeats, STRs, have shorter repeat units, usually dinucleotide or tetranucleotide units. 3. Single nucleotide polymorphisms, SNPs, are variations in a genome with different nucleotides at specific positions. SNPs can be detected using oligonucleotide hybridization, which allows selective pairing between an oligonucleotide and a DNA molecule. Techniques like DNA chip technology and solution hybridization have been developed for SNP screening, which offers faster and more efficient detection than gel-based methods. Limitations of genetic mapping. 1. Genetic mapping does not provide information about the physical distance between genes, as it provides only the outline. 2. Recombination levels can differ across various parts of the genome. Some parts exhibit high rates of recombination, known as recombination hotspots, which can impact the accuracy of genetic mapping. 3. In organisms where obtaining a large number of progenies is difficult, the resolution of a genetic map is limited because it depends on the number of observed crossovers. 4. So, it is important to consider mapping information generated using other methods as well. 5. Various physical mapping methods have been developed to address these limitations. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.